Hello everyone, today we will learn about FASTA file. FASTA formatted genomic files store the sequence information. Here I am showing two examples of sequences. First one is the DNA sequence with A, T, G, C nucleotide bases. Second one is the protein sequence with single letter amino acid codes. We can store this sequence information in a FASTA file. So this is an example of FASTA file. This is a text file. It has two major sections. The header line always starts with the greater than sign, which is followed by the sequence itself. In a FASTA file, we can store more than one sequences. For example, here we have stored two different sequences in a single FASTA file. When we store multiple sequences in a single file, we often refer to it as a multi-FASTA file. So here we can see each sequence has the header line starting with the greater than sign followed by the sequences. We need to remember another thing. In a FASTA file, we only store single letter codes of nucleotides or amino acids. FASTA file format is flexible in many different ways. For example, FASTA file format does not have the strict requirements for how many bases or how many characters there will be per line. For example, here I am showing 20 bases per line and the last line has only 2 bases. In general, we keep 60 to 80 characters per line in a FASTA file. That helps the readability of the file. Also, the header line does not have a formal definition. Generally, the first string in the header line is considered as the sequence ID and rest is considered as the description or name or other information of the sequence. Since there is no formal definition for header line, different databases uses different formats for header lines. This is an example of ensemble formatted sequence. The header line starts with ensemble ID, the species name, protein name, and other informations. At NCBI, we can see a different format for the headers that has the IDs, species information and other information. So we need to be very careful when we are working with data from multiple sources. We need to understand their header format to parse them programmatically. As I mentioned earlier, FASTA file is a text file. File extension in most cases will not matter, but we commonly use .fasta or .fa. NCBI distributes their genomic data in FASTA formatted files using different extensions. Those extensions help to understand what is inside the file. For example, .faa for the amino acid sequences, .ffn for the untranslated nucleotide sequences, FRN nucleotide sequence for RNA related features, etc. There could have several other FASTA file extensions. Here I am showing an example of a multi-FASTA file. We can see there are 3 sequences in this file and these sequences are ordered by the sequence ID 1, 2 and 3. If those were not sorted or ordered, we could sort them programmatically. Also, we can find is there any duplicated ID in the FASTA file. Sometimes FASTA file could contain thousands of sequences, so we need to use some programs to find those occurrences of duplicates. Often we want to know what is the length of the sequences, what is the GC percentage, and how many sequences are there. So those statistics can also be generated using different programs. Often we are interested to look into the specific substring or a motif sequence. For example, here I highlighted the GATA motif, GATA. We can count how many GATA sequences are there in each sequence. Also, we can identify the location of the GATA motif in a sequence. In often cases, we may be interested on removing some of the bases at the end or at the beginning. Those operations can be performed using different bioinformatics tools. Here I am showing some examples of such tools. We can use BioPython's seq.io. Seqkit is a very popular tool to work with FASTA files. FastText toolkit is another alternative. There are a few other tools available to work with the FASTA files. We can perform most of the analysis on FASTA file using the tools. Please check out my other videos on those tools. Let me know your thoughts in the comments below. Thank you for watching this video. See you in the next one.